Hi guys, let's continue our discussion of Chapter 4, Genetic Inheritance, Deviation from Mendelian Inheritance, focusing on sex-linked genes. This is the learning objective that you need to complete at the end of the lesson. At the end of the lesson, students should be able to construct a genetic diagram to show sex-linked genes using hemophilia as an example. Sex-linked genes. Sex-linked genes are genes which are located on the sex chromosome. As we all know, there are two types of sex chromosome in human. They are X chromosome and Y chromosome. X chromosome, as you can see in the diagram, are larger or bigger in size compared to Y chromosome. Due to this, X chromosomes are able to carry a lot more genes compared to Y chromosome. Since females carry two X chromosomes or have the genotype of XX, they are usually less affected by sex-linked condition or diseases. This is because if they are in heterozygous condition, the normal X chromosome will mask the effect of the X chromosome carrying defective alleles that causes the sex-linked conditions or diseases. Since males only carry one X chromosome, they are more likely to be affected by sex-linked conditions or diseases. This is because if they receive a defective X chromosome from the mother, the expression of this defective X chromosome cannot be masked since they only carry one X chromosome. An example of sex-linked condition is the gene for normal blood clotting ability located on the X chromosome. The X chromosome can carry the dominant allele for this gene that give rise to normal blood clotting ability. The X chromosome can also carry the recessive allele for this gene that will result in abnormal blood clotting ability or the condition called hemophilia. When talking about sex-linked genes, there are two angles that you will be looking at. The first angle is the sex of the individual, and the second angle is the condition of the individual. In this case, their blood clotting ability. Individual with this genotype will be a female and having normal blood clotting ability. The individual with this genotype will also be female and having normal blood clotting ability. Even though carrying the defective allele, the presence of the dominant allele will mask the effect of the defective allele, making this individual to have normal blood clotting ability. However, this individual could pass the defective allele and possibly cause hemophilia in her children. So therefore, this individual is termed as carrier. Individual with this genotype will be a female and having abnormal blood clotting ability or the condition of being hemophiliac. The individual with this genotype will be a male with normal blood clotting ability. And lastly, individual with this genotype will be a male suffering from hemophilia. As you can see, females can either be normal non-carrier, normal carrier, or hemophiliac female. But for males, there is no carrier. They are either normal male or affected male. Now let's try build a genetic diagram for this problem. Hemophilia in humans is due to an X chromosome mutation. What will be the result of mating between a normal non-carrier female and a hemophiliac male? A normal non-carrier female will be homozygous dominant, and this will be the genotype of a hemophiliac male. This individual can only produce one type of gametes. Meanwhile, this individual can produce two types of gametes. Fertilization will give rise to F1 phenotypic ratio of one normal female and one normal male. Now, if the question asks what is the probability of the couple to have a hemophiliac daughter, we have to consider the probability of the child to be a girl times the probability of the girl to be hemophiliac. As you can see, there is a 50-50% chance for the child to be a girl, hence 1/2, and there is no chance for the girl to be hemophiliac, hence times 0, giving the overall probability of 0. But if the question asks, what is the probability of the son to have normal blood clotting ability, since the couple already know that they are having a son or a boy, so we no longer take the sex probability of the child into account. So we quickly look at the male, and the son is 100% to be normal. Now let's take a look at another example. This is the cross between a normal female oldog carrier with a hemophiliac male. This cross will result in the production of F1 with phenotypic ratio 1 normal female, 1 normal male, 1 hemophiliac female, 1 hemophiliac male. If the question asks what is the probability for the couple to have hemophiliac son, since we still do not know whether the couple is getting a son or a daughter, therefore we have to take the probability of the child's sex into account. It will be like this. As you can see, there's a 50-50 chance of the child to be a boy, hence 1/2. And amongst the boys, there is a 50-50 chance for the boy to be hemophiliac. So again, 1/2. So 1/2 times 1/2, giving the overall probability of 1/4. But if the question asks, what is the probability for the daughter to be a hemophiliac? In this case, the couple is sure to have a daughter. In this situation, we do not have to take the probability of the child being a girl into consideration. As you can see from the genetic diagram, there's a 50-50 chance of the girl to be hemophiliac. So therefore 1/2, giving the probability to be 1/2.